from TO4.
I'm Amy from TO4.
I'm Stephanie from TO4.
Before we start, let me show you something. That's what I want to show you, a piece of paper. Can anyone tell me what paper usually made of?
Tree.
Yes, yes, you're right, it's tree. And our group knows that genetically modified organisms make news every day, and we are curious about how GM trees interacting with the environment. An experiment of 4-year-long field trial was performed on GM low-hanging trees in order to study the impacts on the environment and the performance towards the lignification as well as the paper production process. And the results show that the GM trees growing in arrays showing not much difference from reference trees. And however, in regard to the lignification, the GM trees perform better, which means that less chemical is needed for less chemicals need to digest the lignin. And in addition, GM trees yield higher quality pulp. To sum up, our group believes that further investigation is needed since there are some limitations about this piece of study, and the limitation will be introduced by Steffi.
So after the short introduction to our group's topic, I will talk about the 4 main points of concern we identified. The first one is the researchers set up two experiments, one in France and one in England, and the French trial was composed of 10 trees in total, and the English one of 24. And both times the trees were surrounded by normal trees or wild trees in order to minimize the edge effects. However, we think that the sample size was too small to generalize the findings towards commercial plantations found in monocultured forests. And furthermore, the trial setup did not prevent the dispersal of genetically modified pollen into neighboring trees, and so outcrossing will most probably occur. And these effects were not investigated. The second point is about the length of the English trial, which was originally planned as a long-term trial, and it was ended premature because anti-GM activists cut down the trees. And due to the short, however, the researchers sampled the destroyed trees immediately to further examination. But we think due to the short experiment, the results may have a limited accuracy, as trees grow very slowly, as you know, and they require long-term trials in order to fully understand the effects on the environment. Our third point of concern questions the annually control of insects and pathogens. We know that the authors did this only once a year because it is recommended by governmental standard survey protocols. However, we think as the trees have an artificial modification, the sampling should have undergone more often in order to obtain more detailed information. And especially because the annual control did not take into account the natural inclining and declining cycles in population size. And another important point is that insects next to the trial are not examined for abnormal changes like it happened in the monarch butterfly population in the US after the caterpillars, here you can see one, fed on the genetically modified corn. And the last point of concern is about the soil sampling after the destruction of the trees. The results were obtained from 6 trees, 6 modified trees, each sampled 3 times in a radius of 30 centimeters. And the procedure we are questioning here is the sample depth, which was only 0 to 10 centimeters. And the researchers inferred from their results that the mycorrhizal community is quite similar to the one of non-modified trees. However, we think that the results may be limited because they only sampled a small part of the entire mycorrhizal community. And therefore, yeah. And now we will pass over to Amy, who will talk about the relevance of the article.
Thank you, Stephanie. And now I will talk about the relevance to the article to our topic. And at the very beginning, our topic is decided to talk about the pros and cons of the genetic modified trees to the environment. And as we noticed that there are too many information and too many data in the internet, therefore we decided low content GM trees. And here is the article we find. And we found this article is quite interesting and useful and suitable for our topic because the author do some experiments go in the field, which is quite different to the traditional way, and which is only do in the greenhouse. And therefore, the GM tree will have interaction with the wind, the rain, also the force, and therefore we can know more details on the GM trees and the environment. And also, the author do the GM trees in a large scale, which means we can obtain more data on the relationship with the GM tree and the environment. And also, the author chose 2 kinds of the common low-living tree. This is the 2 species, as we know that the process of lignification involves a lot of enzymes. And this tree, the first one tree has this enzyme dysfunction. The second kind of tree has this enzyme dysfunction. And therefore we can obtain different information from different type of the tree. And because if the enzyme is not function well, then the lignin produce will become less. And therefore the paper making process will be more efficiency. And also the author choose 2 countries, United Kingdom and France, for the experiment, because different places will have different climates and the soil condition may be different. And therefore, the more information we can get from the experiment. And also, the author choose the duration of the experiment is about 4 years. And although it's still not long enough, but I think it's suitable for a tree experiment because trees go very slowly. And finally, the author measured the goal of the transgenic plants, such as the height, the diameter of the trunk. And we can compare with the GM trees and the normal tree and to see whether the tree will healthily go fast or slower compared with the normal trees. And also, the author measured the biological interaction between the GM trees and the normal trees, such as the insects, the fungi, the bacteria they found in between those normal trees and the GM trees. And lastly, the author also found the microbial organisms in the soil and also measured the decomposition rate of the carbon and the nitrogen, and to see whether the normal tree and the GM tree will have any difference. This is all of our presentation. Now is the Q&A session.